All right, cool. So we're going to get started on the next session. So this is Kat, and she's going to talk about herbal medicine efficacy. Sort of.
Yes, that's good. Thank you. So it's my first talk. Oh, that's very loud. Is that too loud? That's good for you then? Okay. So I'm talking about clinically relevant polymicrobial biofilms in recurrent UTI, and I'm using these to test herbal medicines on. And just to orientate you, it is different to everyone else. It's lab-based research, so it's test tube research. We do it in the B block, level 10. Yes, so play with cells. And so that's me, Cathrina Geldard. So I have qualifications in naturopathy and biomedical science. And these are my two supervisors, Andrea and Jessica. And the next speaker, we share Andrea. So these are really awesome people. So talk to them if you see them. So firstly, because it's a UTI biofilm, what is a UTI or urinary tract infection? Well, first, the statistics that there's over 400 million per year diagnosed and causing over 200,000 deaths. So they occur in 50 to 60% of females, 12% of males, and 5% of children. And even with treatment, they are highly recurrent, so they come back within 6 months. And so that happens in 44% of females and 5% of males. And, it can happen again and again, and 1% of the population actually have a chronic UTI where they've got a low-grade infection all the time that doesn't resolve. And so we say that it's 30 to 50 times more common in females, so this is because of hormonal influences, and also we have a much shorter urethra, which is the tube from the outside of the body to the bladder, so it's easier for things to get in there. And how we treat UTIs is just with antibiotics. So you can get antibiotics the minute you've got a UTI, and the ones with recurrent UTI will get these low-dose antibiotics all the time to try and stop them getting another UTI. And so that means that 25% of our antibiotic prescriptions are for these urinary tract infections. And this is alarming because we have this looming crisis where superbugs are predicted to kill 10 million people a year in the next few decades. This is equivalent to current deaths from cancer and diabetes. And so all our antibiotics for UTIs are majorly contributing to this crisis. And also antibiotics have some side effects, such as giving you nausea and vomiting. Some cause kidney toxicity, and they also cause imbalance in our you would have heard of your gut microbiome, which is really important for your overall health. And when you have recurrent antibiotic prescriptions, it just makes this problem worse. So this is actually a UTI, and so it's an infection of the bladder or kidneys. And so it's only the kidneys in 1/3% of the time, thank goodness, because that's more serious, because it can spread to the bloodstream from there, and it's a systemic infection. So a UTI, as I said, occurs when bladder goes up the urethra. So it usually comes from contamination from the anus, and the bladder gets in there and colonises and makes an infection. And the biofilm that I've circled is definitely a large part of the disease process, and this contributes to why antibiotics fail in treatment, and I can tell you why. So this is a biofilm, and so rather than the free-floating bacteria we see at number 1, these planktonic ones, they're free-floating in a liquid environment. This biofilm is a structured community. And so the bacteria attach to each of the surfaces and each other, and they build this matrix, and they coordinate their behaviour through talking to each other with their special bacterial language. And so as the life cycle progresses, it gets more complex. There's different relationships within, it has different properties, and all this, makes treatment complicated. And this shows how antibiotics don't work. So we've already said that we treat with antibiotics. They work by killing the bacteria or stopping them reproducing, but they don't work very well in this biofilm environment compared to those free-floating cells. So 10 to 1,000 times the antibiotics are required for this kind of infection, which means you might not even be able to get that amount into a human being. So it makes biofilms sometimes very difficult to eradicate. So this picture is showing why. So the big one is the line around the outside, the protective matrix they make. And so that protects all the bacteria inside, not just from the host immune system, but it makes bacteria protected from antibiotics because it's hard to penetrate. But also there's just different little pockets in there of different nutrients and oxygen and pH levels that make different microenvironments. So the antibiotics won't work the same amongst all the bacteria. And even some slow down their growth rate. So that's what antibiotics target, is rapidly dividing cells. What else? They even share genetic information, including how to evade antibiotics. So there's a whole bunch of reasons there why the antibiotics don't work. But what is interesting is then those can be some targets from alternative treatments. So to make things even more complex is that urinary tract infections, or UTIs, 35% of the time they actually involve more than one bacteria. And so these polymicrobial or mixed species biofilms are even harder to treat. So it can be because the bacteria that works against one of them doesn't work against the others, but also these bacteria can work together to improve their survival. And so that means the infections can be more severe and it makes antibiotics even less effective. They have more resistance. So most research so far has been on those single species biofilms, not much with multiple species, definitely not in the UTI context. And also until recently, we wouldn't even admit that a UTI could be caused by multiple pathogens at once. We would think just the test sample had been contaminated. So there's not a lot of research about it. So I'm trying to change that, obviously. So this is a very oversimplified way of how I make a biofilm. And so I'll talk about why I've chosen those three. I'll call them bacteria. One's not, but you're not microbiologists, so I don't think you care. So the first one, E. coli, the yellow one, is the causative bacteria in at least 65% of UTIs. And the second one is often quoted as the second or third most common cause, depending on the patient population. But more importantly, if it is in a mixed species, so multiple species infection, it's usually with the first guy. And this third we'll call bacteria is actually something that's really commonly everywhere in the body, doesn't cause any symptoms, but unless it's in too high amounts, then it can cause thrush or UTIs. And these too high amounts can be seen in people who've had multiple antibiotics, like recurrent UTI. But I put less of that little guy in the biofilm. And so when they're in the tube, we grow them in that free-floating state. We're just trying to get more and more of them. So grow them overnight in an oven that's the temperature of a human being. And then we put them in a little plastic plate. And then 24 hours in the oven again. And then when they come out, I dye them purple. And so what I'm measuring here is, number one, how purple the biofilm is, and that's telling me how much of that protective coating, their matrix that they have. So the more purple equals more matrix. And I measure how many live bacteria go into the plastic tray, so in their free-floating state, and how much comes out as a biofilm. For two reasons. So we know that only a few % gets incorporated into that actual biofilm. But also, these measurements are what I'm going to measure against later when I look at herbal medicines and see that I've reduced the biofilm. So these are pictures of my biofilms, and you're not expected to know what it is. So all I want you to see is that I have all these different biofilms there. Some are one species, two species, three, and you can see that the amount of purple is not the same. So no two biofilms are the same. And my data here backs this up. So this is actually counts of, like I said, I count the live bacteria going into the plastic tray, how much comes out as a biofilm. And so with my three species, we can see that in the bottom left picture, my number one pathogen is my E. coli going into the biofilm, but coming out, then the green line is now the number one. And when I put this as a graph as a percentage of the whole, it's even more obvious that in the first one, my E. coli is 73% of the total, And in the second one, my efficacy is now the major player. And my little tiny, as I said, my happy one that doesn't usually cause much symptoms is only a small amount in the biofilm, but we can see next it still plays an important role. So this is my 3 species, but now made in combinations of 2. So they're 2 species biofilms. And what we can see in this bottom right-hand corner is that my 2 main ones in my triple species now have a different relationship. In this one, it still starts off with my E. coli is the dominant one, but coming out of the biofilm, it's still the dominant one instead of it swapping to the E. faecalis. And so when we look up in the top left picture, so these are the ratios of what's turns from free floating into the bacteria, and you can see that they don't actually make a very good biofilm together. But what is interesting is the big blue line on the very left, my little tiny bit of candida that's in there, it's much happier in its relationship with one of the bacteria than the other. You can see it incorporates a lot into the biofilm and not in the second one. I hope you can see that. Anyway, so it just shows us that back to the biofilms are really different depending on what species of bacteria is in there. So all this is supposed to highlight is that we really can't extrapolate knowledge about one biofilm to another context. So, yes. One? Okay, good. So what am I doing with the biofilms? That's the important thing for me, because I'm testing herbs. How am I going to find the herbs? So I'm using this methodology, and it's my primary supervisor came up with it. So we're looking at traditional medicine and contemporary practice, coming up with some herbs. And what I'm doing is some mechanism of action studies where once a herb has antibiofilm action, I'll be working out how specifically. But this could be expanded to do drug discovery and then onto clinical trials, but I'm not doing that. So I've done my scoping review, and the traditional evidence I looked at was the Balkan region, because there's high biodiversity, they've got huge amounts of plants. They also use herbs a lot in their primary care, so in the community they use a lot of herbs. And I also looked at naturopaths because they prescribe herbs day in and day out clinically. So I came up with 121 target herbs that are used specifically against bacteria, but we did get 175 more that are used for other parts of UTI, like improving the host immune system, reducing UTI symptoms such as pain. I also did an international survey of naturopaths, so asked them what they use clinically now, and got a very poor response rate. Even so, we got 7 herbal medicines, and those 7 are all in my scoping review. So hopefully that means that these are more likely to work. So I said I'm doing mechanism of studies next after I've tested my herbs. And so this is just to show you that, so I said that antibiotics work by killing the bacteria, stopping them reproducing. And those are really easy ways for bacteria to learn to adapt to and evade. So these are examples of ways that other herbs have worked in other biofilm. So we just saw in that biofilm life cycle there's multiple places we can intervene to disrupt biofilm. So we could stop it attaching to the bladder cells in the first place. We could stop it making its protective coating. We can make it disperse so that the antibiotics can kill the free floating one. We can interrupt the bacteria talking their special language and coordinating their behaviour. And we can even use them alongside antibiotics so that the bacteria can't make these little pumps that pump the antibiotics out of their cell. So all of this, also herbs can do all of these at once as well. So that makes it a pretty good option for our antimicrobial resistance. And to sum it up, just to show, so because it was my title, that we're trying to improve our clinical translation. So when we're doing this test tube research, it's not well extrapolated to humans, so we're doing our very best. So we've made our biofilm polymicrobial, so that's relevant for some UTIs. Next, we're actually going to grow them on bladder cells, so that's even more like a human infection. We chose our herbs by asking what people are using, so not only will that increase our chance of successful target, but also those same clinicians could use our mechanism studies when we do them, and that might help them refine their treatments. And this biofilm I'm actually doing today, my first time, I'm testing antibiotics on it that doctors actually use. So I can compare my herbs to the antibiotics. So that makes it clinically relevant as well. But other people could test nutrients and probiotics and all sorts. So all that should theoretically lead to better clinical translation of our preclinical results. Thank you for listening. What do I do? Is that it? Did I talk fast enough? Okay.
Wow, that was really, really awesome. Thank you. That was just incredibly interesting and cool.
Oh, is it? Okay.
Yeah.
I'm a bit patronising anyway. Okay.
And yeah, obviously a little bit terrifying. The resistance. No, that was awesome. Thank you.
Okay. Okay.
Thank you so much for that. I work a lot with, I'm in the hospital and I work a lot with people that have UTIs, dementia and delirium. So it's really important work to get to the bottom of it.
Yes, and under-researched. Yes.
Very much so. So I guess my question is just, and you might have explained this, but just to make it a little bit more simple terms for my brain, is this going to be a prophylactic solution, or is it like instead of a Hyprex type medication, or is it instead of antibiotics, or is it both?
Well, at first, all those options are open. So I've done a bit different to other people who've already finished their results. I'm smack bang in the middle of this because I wanted to present something that was different. So I'm nowhere near a paper with this. So as I said, today I'm testing its antibiotics. So all I've done is make the biofilm. But ideally, I want a herb that works, a disrupting one, because that's where there's a really big gap. Instead of stopping it forming, we want to break down one that's already there. So ideally a treatment, but prevention is second best. Working alongside antibiotics would be the next option. Hyprex, that does work. So it does work in some cases. So like all UTIs, so it depends on the biofilm, on the pathogen. So Hyprex works great sometimes, but I think you have to have quite a alkaline environment. Anyway, I have seen it. Anyway, so this will work on some UTIs, whereas Hyprex would be good on something else. Okay. So ideally, I want the herb alone, but that may be too idealistic.
Something to work towards. It's great. Thank you.
Any other comments? Oh, yeah.
Yeah, you would have heard cranberry. Everyone takes cranberry. It's good for it. When it works, it works. It's not good for every UTI.
No, I actually stand by Pam on that one. Like, I love science, and that was fantastic. So, and I think it's a really great project. And I've heard a lot about UTIs on the radio and on the television and stuff like that. And I know it's a very intractable problem. So you're on a winner there. My question, I suppose, is, my understanding, correct me if I'm wrong, but after the survey of practitioners, you'd sort of narrowed the field down to 7 possible contenders.
Yeah.
So if you're doing a study on that, that's a lot of variables. You've got 7 possible active agents, then alone and in combination with antibiotics. That's a lot of variables.
Yes.
Are you going to start out exploring all of them, or are you planning perhaps to narrow the field down?
I literally had to pick my herbs yesterday, the starting ones. I've got to admit, I was really idealistic, so I haven't mentioned that there, but I'd actually even love to use herbs as we use them in naturopathy with multiple together. Yeah. That makes it very tricky, because usually in this sort of research, it goes sort of down a pharmacy route where they isolate even specific constituents from a plant. Okay, that's easier. But I'm ideally hoping that someone can not even have to buy a supplement, drink a herbal tea. That would be awesome. So yes, we are limited by what we can access as well. So a lot of my herbs came up, we can't get them easily to test. So I've got a list of about 7. Yes, that does add up to a lot. But yeah, it will be a lot of work if nothing shows activity and I have to then start mixing them together, mixing with antibiotics, using them at different stages of the biofilm. But I am hoping, like, we are asking people what they're already successfully using in a person clinically. Yes. So that should increase my chances of success. But just because it works in a human doesn't mean it works in a test tube. So if it doesn't work here, it doesn't mean it actually doesn't work at all. Yeah. So it's just part of the evidence, and other people will do something else, I'm sure.
Well, I had to have you back next year to sort of present some results.
Yes.
We've probably got time for one more question, if anyone had any.
Yes. No? Okay. Yes.
I don't have a question, but I
Okay.
did want to compliment you on your beautiful slides and how clear they were to understand. Science is not my thing, but I did find that your slides and the way that you've communicated what I consider quite complex information really clear. So I just want to congratulations on that.
Thank you, because I found that a bit hard. I thought they're not microbiologists, but you don't want to be patronising. So it is for my first talk. I thought, where's that line? So that's good.
That's good. Good feedback. Yeah.
Thank you.
Thanks. Well done.
Thanks.
Thank you very much.